Structure of the eye. So the eye is made up of a retina, and that retina contains lots and lots of photoreceptor cells. There are two main types of photoreceptor cells. Those are the rods and the cones. The rod cells synapse onto bipolar cells in a ratio that means you have spatial convergence. And what we mean by that is 2 or 3 rod cells will converge onto one neuron that then feeds into the brain. Whereas with a cone cell, that feeds one-to-one to the neuron that goes to the brain. Now there are 3 types of cone cell. Each type of cone cell, you have a red pigment in one of them, you have a blue light-sensitive pigment in another, and green light sensitive in another. So effectively what happens is, if green light hits this particular cone cell, then the pigment is really sensitive to it, it breaks down in vast quantities, and so you get an impulse being sent straight to the brain. The brain can then interpret that information and understand that there's a green light coming from this particular part, because the light that entered the eye hit this particular part. We find that the cone cells are concentrated in an area of the brain called the fovea. The rest of the retina is mainly rod cells, so that means that our peripheral vision is quite sensitive to low light, but it's no good at picking up colour. So if you held a colour pen in your periphery, you wouldn't be able to tell what colour it was, but if you hold a coloured pen in front of your eyes, because that's hitting the fovea, so your lens is able to concentrate the light that enters your eye onto your fovea, so anything that's in front of you, you can actually see in really good colour, and you have a really good resolution of that. You notice that in the dark, where you don't have much light available, everything seems pixelated, and that's because it's your rod cells that are able to respond to that. And the reason why things seem pixelated and there's not very much good resolution is to do with this spatial convergence. Basically, because the light entering, so the light enters the retina in this direction, you don't know which of these rod cells it's actually hit to cause this one cell that's going to the brain to fire the impulse. So because of that, things seem a little bit less accurate. So we lose our acuity because of our resolving power is poor with the rod cells. Whereas with the cone cells, we know exactly which cone cell was hit by the light that came into the eye, because each cone cell synapses with a separate cell that goes to the brain. And so we can really highly resolve the light that comes in and hits our cone cells. So that's to do with spatial convergence and visual acuity in the eye. When we look at what's happening at the level of the synapse, then we can see that in the level of the synapse, all that we have is, if you have one cell synapsing onto another cell, the action potential reaches the presynaptic knob, and as it reaches the presynaptic knob, it causes depolarization of the membrane that opens your calcium channels. Your calcium ions diffuse into the presynaptic knob, and as the calcium diffuses in, that causes vesicles to be moved and fuse with the presynaptic membrane. As they fuse with the membrane, they release neurotransmitter which diffuses across the synaptic cleft. It binds to ligand-gated sodium channels, so basically the neurotransmitter binds to the channel protein opens, sodium rushes in, that causes depolarization of this membrane, which causes voltage-gated sodium channels to open, and the action potential continues. Now, if you have one cell and it only releases a tiny amount of neurotransmitter, it might not cause enough sodium to enter and cause depolarization. So what happens in the case of the rod cells, because they're not releasing enough neurotransmitter, there's multiple rod cells. So that allows tiny amounts of light that hit each of those 3 rod cells to release enough neurotransmitter onto this one cell to make it fire. And that's why you're able to see it in low light, because even though there's a tiny bit of light, because they all converge on the same postsynaptic neuron, it's possible to trigger an action potential in that postsynaptic neuron and get yourself an action potential and still be able to see things. But you won't be able to see it in light, because the amount of neurotransmitter released when a tiny amount of light hits a cone cell isn't enough to trigger an action potential in the postsynaptic neuron. So that's why we see in black and white, in low light, not colour.